So do you want to start?
Yeah. Just tell me when to
Go.
Okay, my name is Emily Ferguson,
And I'm Hannah Cass.
and our enzyme for our project is beta secretase 1 or BACE1. So why do we choose BACE1? Well, it's found in the human brain, and it's also found in many other species such as monkeys, rats, mice, etc. It has been linked in many studies to Alzheimer's disease. Now, as we know, Alzheimer's disease occurs when people start forming plaques and tangles in their brain. Mild to moderate symptoms of Alzheimer's include memory loss and cognitive difficulty, and can even include hallucinations and paranoia. Severe Alzheimer's, as you can see in the picture, actually ends up leading to atrophy of the brain and unfortunately eventually death. So Alzheimer's is a very tragic disease, and this enzyme is heavily studied to try to find ways to reduce the prevalence of enzymes in elderly people. So now that I've talked a little bit about the introduction, Hannah is going to talk a little bit about the structure of our enzyme.
Okay, so in talking about our structure, we're going to start off with the primary structure. This specific protein has 510 amino acids in its sequence, and then the primary structure gives way to the secondary structure map, which is going to be here, but we're having formatting issues right now. So when it will be here, you'll see that it's a mainly beta protein, so there are a majority of beta sheets, and then there are also a few alpha helices and a number of turns connecting those beta sheets. It is a globular protein. And so here's the tertiary structure. The important thing to note about this is right here is the active site, and that's where that amyloid precursor protein goes in. And you can see here, like the beta sheets, this kind of looks almost like a beta barrel. And here are the alpha helices, although the specific tertiary structure of this protein has not yet been identified. So what does all this protein actually do?
Okay, so as Hannah started mentioning, our enzyme actually cleaves a protein known as amyloid precursor protein. Now, there are several forms of secretase. Alpha secretase is also found in the brain. It cleaves APP, amyloid precursor protein, as well. However, when it cleaves it correctly, if you will, and it actually, the result of that is actually neuroprotection. It's good for our brain for it to be cleaved in that manner. However, if beta secretase and also gamma secretase, another very similar enzyme, cleave amyloid precursor protein, you get amyloid beta protein, which is a very bad protein to have in your brain because it causes a calcium imbalance. And because calcium is so important for neurotransmitters and sending signals and active potentials and stuff like that, if you have a calcium imbalance, that can be detrimental to the brain. So it makes a lot of sense that an improper cleaving ends up leading to Alzheimer's.
And that amyloid beta protein is what causes the plaques. So what is the mechanism, Emily?
So the mechanism isn't super known. There isn't an actual arrow pushing mechanism. More is actually known about the mechanism of gamma secretase. But for beta secretase, what we do know that what it does, it cleaves APP at one certain point, and then gamma secretase cleaves it at another point. This is a good picture because it shows that APP is a transmembrane protein, and it cleaves it right in this area. It's also surrounded by this protein, which name I do not know right now, but I will know later. And we will know further details of the mechanism in our final presentation.
So now that we've taught you guys about kind of the background of our protein, we're going to go into a bit of primary literature. So the article that we're going to be presenting is beta site amyloid precursor protein cleaving enzyme 1 increases amyloid deposition in the brain parenchyma but reduces cerebrovascular amyloid angiopathy in aging mice by Willem et al. So first, what they did was they took a series of mice and they inserted the base 1C DNA into their genome so that it could be expressed. And then they looked at a Western blot analysis and ELISA analysis, and they did immunohistochemistry, which we went over. That's where you use the two antibodies to kind of quantify the amount of protein that was in these mice. So this is kind of showing where they put in the cDNA. So right here where they have that amyloid precursor protein, they also put in the base cDNA so that they would be, is it constitutively? No, they'd be promoted together because they have the same promoter region. So when you make amyloid precursor protein, they'd also be producing that base protein. And then on the bottom, you can kind of see just like that's them showing how much a protein is actually being produced and the size.
These are some more results from the article that we will also discuss in further detail. It shows prevalence of APP and it shows prevalence of amyloid beta proteins, so we will discuss that further as the results of the experiment.
Yes. So, but you can kind of see, especially from this one, they're looking at the amount of amyloid beta peptides, and it's higher, the amount of protein is higher in those that were treated with the base cDNA than those that were not. And that's kind of just a blown up image.
Good.
So this is, yeah. Cool. And I think this is also, I think this is Western blot.
Okay,
Yeah.
good. And we have significant p-values down there, so we will discuss what is significant.
Yeah.
Yes. And as a conclusion, Alzheimer's really sucks. A lot of research is being done right now. Neuroscience is a very hot field to be getting into, and a lot of research is being done on neurodegenerative diseases in general. We do know quite a bit about beta secretase and its possible role, but there is quite a bit more research to be done in the mechanism and exactly what it does, so that we can find a way to stop it and inhibit the protein. Yeah. Okay. I'll stand here.
Wait, I feel like, do you know how to work YouTube? Do you know how to
No.
cut it off? Okay. It's not stopping. Why aren't
I think it
you
did stop.
stopping?
No, it did stop because it said, I think it was stopped, wasn't it?